Hello guys, today we will learn about blood. What is blood? Blood is a connective tissue in fluid state. It is liquid, in liquid form. This is what is blood. Blood is a connective tissue that is in fluid state. Now, what is blood volume? Blood volume is usually 5 liter and you may say that is 8% of body weight or 80 milliliter per kg body weight. Suppose a person is of 70 kg, then his blood volume is 80 into 70. That is itna liter. If 60, then it is multiplied by 60. It is not fixed, but 5 liter is taken as normal. Now, composition of blood. Blood is formed by two things. Plasma, that is liquid form, plasma is 55%, and hematocrit, that is cellular part, is hematocrit or packed cell volume is 45%. Hematocrit or packed cell volume is 45%. Now, cells. Cells are known as blood cells, or there are three types. Erythrocytes, red blood cells, or better to say red blood corpuscles. And second is leukocyte, WBC, or white blood cell corpuscles. And third is thrombocytes and or platelets. This is three types of cells are present in blood. Now, concentration of or number of cells. RBC is 5 million per cubic liter of blood. 5 million per cubic milliliter of blood, macroliter, 5 million per macroliter, or it may be said per cubic milliliter, or in another words it is 5 into 10 to the power 6, 5 into 10 to the power 6 per macroliter. Leukocyte or WBC is 4 to 11,000 per macroliter, or 4 to 11 into 10 to the power 3 macroliter. And thrombocyte is 1.5 to 4 lakhs per cubic liter, or 0.15 to 0.4 in 10 to the power 6 cubic liter. This is about blood cells. That is, blood cells are of two types: erythrocytes, leukocyte, and thrombocyte. Erythrocytes are also known as RBC, leukocytes are known as WBC, and third is platelet. Thrombocytes is known as platelet. Usually this is the normal in use in its platelets. Now, erythroid concentration of RBC. How much RBC is present? That is 5 million per cubic macroliter. Per macroliter, or it may be said cubic milliliter. That is, WBC is 4 to 11,000 per macroliter, and thrombocyte is 1.5 to 4 lakh per cubic liter. You have to, in another word, it is, nowadays this is written, that is 5 multiplied by 10 to the power 6 RBC per macroliter, 4.11 to 10 to the power 3 macroliter, and 0.15 to 0.4 in 10 to the power 6 macroliter. Now coming to plasma. Plasma is fluid portion, and it is 55% of blood. That is, when we allow to stand this blood mixed with what? Mixed with anticoagulant so that it will not coagulate. Then you will see that 45% will settle down. These are cells. Because cells are heavier than the plasma, this will settle down. And here it is plasma. In between them, a thin layer is present that is known as buffer coat. This buffer coat consists of RBC, platelet and WBC. This contains, buffer coat contains what? Number one, WBC, leukocytes and platelet. Now about plasma. Plasma is 55%. That is, plasma is 55%. Composition of plasma, it is 91% is water, 9% is solid. In solids, we have 1% inorganic molecules and 8% organic molecules. Inorganic molecules consist of mainly extracellular sodium chloride, calcium bicarbonate, and mainly intracellular ions are potassium, magnesium, copper, and proteins. In organic molecules, plasma protein is 7%. And non-protein nitrogenous sugar, fat, enzymes, hormones are 1%. This is 1%. Now plasma protein is of different types. Main plasma proteins are albumin. Plasma protein concentration is 6.4 to 8.3 gram per deciliter. 6.4 to 8.3 gram per deciliter. Albumin is 3 to 5 gram per deciliter. Average 4.8 gram per deciliter. Globulin is 2.3 gram per deciliter. That average is 2.3 gram per deciliter. And fibrinogen 0.3 gram per deciliter. This is main. These are main plasma protein. In addition to that, there are many prothrombin and other proteins are there in very less amount. Okay. Now we will deal with plasma protein in next video. And functions of blood: carrier of oxygen, gases, carbon dioxide, all gases are carried by this blood. And food, digested food materials, amino acid, fatty acid, glucose, hormones, enzymes, everything is being carried by this blood. Then excretory function, it will also carry the urea and other things so that it goes into the kidney and their filtration will occur and blood is pure. And this is excretory function. Homeostatic function, it controls the internal environment of the body. Then it acts as buffer. Then osmotic pressure, exert osmotic pressure by virtue of plasma protein. Protein reservoirs, viscosity, and these are the main functions of blood. And in addition to that, we will learn in plasma protein in our next video. One question is very important, usually asked: what is the difference between blood and plasma? Plasma is blood minus cells. Form elements of blood or cells, RBC, WBC and platelets are removed. Blood minus cells is plasma. Then what is serum? Serum is plasma minus fibrinogen and clotting factors 2, 5 and 8. When it is allowed to clot, when plasma is allowed to clot, it will consume fibrinogen and some clotting factors 2, 5 and 8. Other factors are present in the serum. This is the difference between plasma and serum. This is all about this composition of blood. In next video we will talk about plasma protein and then erythrocyte and leukocyte platelets, etc. Thank you very much.